Welcome everyone. My name is Zack Frederick and I work at the Canada-Manitoba Crop Diversification Center in Carberry, Manitoba. That's CMCDC, and I'm your friendly applied potato research agronomist here to talk to you about our research results for the upcoming season, and including stuff that we've had previously. To start with, I'd like to talk about our overall variability study. This was started in 2016 with the goal to identify what causes low yield in parts of fields and then rank these issues across all of Manitoba processing potato production. It's important to note for what we'll be talking about today that these issues are what's averaged across all of Manitoba's production, all the fields we visited through all the years that we have visited. There will be individual problems like a catastrophic snowfall in September or a localized early blight outbreak, for example, that would be a more significant problem for, say, your individual field, but wouldn't be the same for all your peers across Manitoba. The long and the short of what we do is we record soil and plant nutrient status, soil moisture, and we quantify the presence of disease-causing organisms and pests. And without laboring into the statistics too much, we've looked at approximately 50 of 98 variables across many different yield parameters such as total yield, dollar value, size profile, specific gravity, and generally there would be about 50 variables that would be considered statistically significant that we can associate with yield variability. And then of these 50, we've ranked them in order of significance. So we have a top 10 list or a top 3 list that I'll share with you today of what we consider the most important issues. And it's important to note that this data was created over the course of 5 years using 23 fields, and we keep adding to it every year. The 3 most significant sources of yield variability that we found so far start with verticillium wilt. And specifically it's the propagules of verticillium found in the soil called mycosclerotia. And as the number of propagules increases, especially above 30, the yield of over 10-ounce tubers really drastically decreases. And once you start having hundreds of mycosclerotia per gram of soil, the size profile over 10 ounces basically completely disappears. And this is fairly logical given what we know about the early dye complex. Number 2 on the list is going to be too little soil and impedial nitrogen and row closure. Row closure, for those that keep track of these things, is going to be approximately early July, about 2 months after planting, or nearly 300 P days in years like 2019. At any rate, at this key juncture of row closure, the percentage of yield of 6 to 12 ounce tubers really appears to decrease when the soil has less than about 120 pounds of nitrogen, based on our statistical association. Third on the list of what's important would be too little soil sulfur at row closure. And here it's the percentage of yield that falls in the 6 to 12 ounce tuber size category, the overall sweet spot, appears to decrease when the soil has less than 30 pounds of sulfur found in it through a soil test. This study is important because it is used to scientifically justify further research into treatment plans because we are approaching them, each problem in the order of significance that we've reported. These results are outlined in much greater detail at our Manitoba Potato Research website at mbpotatoresearch.ca. For you, the listener, this study is useful because it identifies problems that you can address across your entire farm because we've observed them through Manitoba, regardless of what particular farm we visited, soil type, and many other factors that would vary farm to farm. This study is also going to be used this year to calculate the average dollar loss associated with these problems when they occur separately or together. So we talked about, for example, with the verticillium wilt, a difference between a soil test of 30 mycosclerotia and a soil test of 100 or 200 mycosclerotia. It creates a different situation. So we're hoping to attach a dollar value to that of how much you lose on average per acre with these kinds of problems. These calculations will then be used in our subsequent studies when we develop treatments. Because we now know the average loss that you would incur, we could then compare it to the average gain that you would return by having a particular treatment over a certain period of time. Thank you, and I look forward to having your feedback on further videos.